Our next speaker is Dr. Christopher Shade. Dr. Shade earned both his BS and from Lehigh University in environmental and aqueous chemistry. He earned a PhD from the University of Illinois, studying environmental mercury. During his PhD work, Dr. Shade patented analytical technology for mercury speciation analysis and later founded Quicksilver Scientific in order to commercialize this technology. Dr. Shade has turned his focus to analyzing how the human body responds to mercury exposure and toxicity. He has developed clinical analytical techniques for measuring exposure levels and has created products that remove metallic and organic toxins. Please join me in welcoming Dr. Chris Shade.
I'm really happy to see where the discussion has gone in our approach to taking mercury out of dentistry. When I got into this business, if you remember the Blues Brothers lines, what kind of music do you have here? We got both kinds. And so you go to your doctor and you say, what kind of mercury detox program will you have? We got both kinds. We got DMPS and we got DMSA. And mercury toxicity is a chelator deficiency. And there is a lack of the whole discussion of the mechanism of toxicity and the way that the body deals with toxicity. And so I started beating the glutathione drum several years ago, and it was great to hear it beat over and over and over again this morning. And we talked, Dr. Ashner talked about mechanisms of chemodefense, and that's what I usually call it. He talked about Nrf2 upregulation and how it brings up all the defense mechanisms for glutathione, or for mercury, by binding mercury to glutathione, restoring the repair abilities inside the cell. And importantly, he talked about energy generation. And so he talked about cells, if you gave them enough glutathione, they could handle the insult. If you gave them enough energy in the form of the NAD, they could deal with the insult. Now, why was that? Because all of these reactions that generate glutathione, that feed the transferases, that link glutathione onto mercury and pull it off of the proteins, all of the transporters, all of them feed on energy. They need ATP generation to do what they do. And so I'm really happy to see where this whole discussion has gone and the very intelligent design of chelators that don't upregulate free radical activity, but downregulate it. And so I want to talk about not a cellular level, but now a whole body level. So I often lecture about microcosm and macrocosm. And microcosm is more these cell culture studies. What does this cell need to survive? And if the cell's got what it needs to survive, it's constantly exporting, pushing mercury out to the periphery, pushing it into circulation. But where do we go from there? We have to push it out. We need to get it out through the liver into the bile tree, out into fecal excretion, or out through the kidneys, through the proximal tubules into the urinary flow. So most of this talk, I'm going to hit a couple of points around systemic programs for detoxification, but I'm going to focus most on the liver and the pathways of the liver and how they backfire. And when you look at these pathways and how they all line up, this is super important because it describes all of the negative symptoms that we get during detoxification. It describes our systemic failure to detoxify. It's not always what were the genes you were given, what are the SNPs, epigenetic, secondary downregulation of chemistry or blocking of the drainage and excretion chemistry is a big, big deal. And we're going to look at the things that drive those blocks and how we can reverse them. So we're going to talk a little bit about toxin pathways. I like to describe them almost every time. It's like it's our map for how we're going to talk about detoxification. Then we're going to talk about liver gallbladder support. And very importantly, it's the movement from inside the hepatocyte through what's called the canalicular membrane. That's from the liver cell into the bile tree. The bile canaliculus is the first collector of bile. It moves down through the bile tree into the gallbladder. And that movement from inside the hepatocyte to out into the bile duct is the most crucial thing that we can look at in systemic detoxification. Why am I just talking about bile? Because the same transporters that are moving bile are moving the toxin conjugates. And when you block the bile flow, you block the toxin flow out of the system. And where does it go? It gets shuttled right back into the blood. That's recirculation or redistribution. And then we'll talk about binders. So what we're going to discuss is how we dump toxins from the liver into the bile, into the gut, and then collect them. And we call it push catch, dump catch, dump catch, dump catch. It's really simple, but we have to know all the rules. So again, just to reiterate this microcosm, macrocosm idea, in the wake of Michael Ashner's talk, I want to bring up this little paper up there that you'll never see, but I've talked about it a lot before. And it was a cell culture study. And this was, maybe 15, 20 years ago. It was before we've had all these things like Nrf2 described. And Nrf2 is sort of a catch-all upregulator that brings a bunch of the glutathione system all up at once. And then they were just looking at little aspects of it. And in this paper, they found cells that were they had a high immunity to metals. So they were very resistant to metals. So we don't want to talk about just net toxicity. We want to talk about resistance and susceptibility. So these cells were very resistant to metals. And when they took them apart, they found that they had high glutathione synthesis, high glutathione S transferase activity. That's an enzyme that links together the mercury and the glutathione. And they had high activity of the transporters that are shuttling the metals out. So these cells were able to push away, always pushing the metals away out of the system. And if they went into the cells and they downregulated any of those processes, the cells were no longer able to do that, and they lost their resistance, and they then became susceptible to the poisoning. All right, so that's at a cell level, and that's invoking all of the glutathione genes and invoking metallothionein. That enables that resistance. But then at a microcosmic level, what have we done then? The cell pushed it into circulation. Now we got to do something with it. We got to get it out. And so that's the macrocosmic side. And here's a picture of the liver draining into the bile ducts, into the gallbladder. This is a diagram we're going to examine. This is all the different directionality in the hepatocyte, in the liver. So think about liver directionality on a macrocosmic scale, and think about resistance on a microcosmic scale. And there's a little boy showing us macrocosmic detoxification. All right, so recall that detoxification has 3 phases: phase 1, phase 2, phase 3. We don't talk so much in metals about phase 1 because it's not necessary for metals. It's necessary for big organic molecules, endocrine disruptors, plasticizers, DDT, hormone molecules. But phase 1, you're kind of nicking the compound and making it reactive, making it bleed a little bit. So in phase 2, you can link it. You can put glutathione onto it, glucuronic acid, sulfate onto it. That makes a toxin conjugate that your body, because of what you conjugated onto it, it is recognized by a series of transporters called phase 3. And these transporters take it out of the cell into the extracellular space. Then it goes into blood circulation. Transporters in the same family pull it into the liver. And then same transporter family pushes it into the bile flow. Exact same set of transporters working at the kidneys. It's called OATP, organic anion transport peptide, pulls from the blood into the renal proximal tubule cells, or in the liver, it's pulling into the hepatocyte. Then the dump out is called MRP2, dumps out either into the bile flow or into the urinary flow. So it's an analogous system in the liver and the kidney. And so if we look at the flow through a system, if I run my green line around here, that's the cell membrane. Above it, you have phase 1 and phase 2 reactions. And then you make a conjugated toxin. It gets dumped through the cell membrane into circulation. There, from circulation, it's pulled into the hepatocyte or pulled into the renal proximal tubule cells, and then from there it's dumped either into the GI tract or into the urinary tract. So that's when everything is working well. I've shown this schematic for a long time, but now we have to add in some of the other pathways that are really important to look at for our story of when things get blocked up. Here's the first pathway that we look at. This is the liver backfiring back into the blood. There is a door out of the hepatocyte into the bile, and there is a door out of the hepatocyte back into the blood. You can call that door the pressure relief valve. The hepatocyte is in charge of handling a lot of toxins, and if it is not able to take dump, it's suddenly, like if the dump's broken, it's just take, the thing's built up, it's all frenzied up, dumps it back into the blood. The pressure relief valve. It's there for bile salts, which when bile salts build up because of cholestasis, they digest the cell, and it's there because of toxins. When they build up, you have to dump them back into the blood or the hepatocytes will die. All right, so we have the pressure relief valve from the liver back into the blood. We have enterohepatic circulation. That's when you dump into the GI tract, but you resorb it back up. Methylmercury is famous for this. It's like a 95% resorption. Cadmium is good at that. Mold toxins are really good at that. I also want you to think not so monodimensionally. This is not a world where there's one toxin called mercury. There's a soup of toxins out there. And when our detoxification systems are broken, the soup all comes into attack. And there's synergies between toxins. Some are additive, some are synergistic. There's toxins that will block part of the glutathione system, and that will allow you to bring more mercury into the system. The mercury then downregulates the immune system. The immune system allows infection. There's a cascade of results, one to another, that is creating the chronically ill person, or even just the weakened person. So we got to think about a lot of things going on at once. And then the last thing is uptake of endotoxin and different dysbiotic toxins. Endotoxin, or lipopolysaccharides, these are parts of membranes of gram-negative bacteria, and they're serious inflammagens. Remember that name, inflammagens. They generate inflammation because your immune system has exquisite pattern recognition for them. And when it senses them, it's worried that there's a septic type of systemic infection, and it upregulates a very severe inflammation to deal with it, to try to save you from this infection. Thing is, there is no infection. It's just little pieces and parts. So endotoxin, we're going to see, is the primary thing blocking detoxification and blocking the bile flow. So it is shutting things down for us. At the same time, it is one of the most significant generators of neurological inflammation, neuroinflammation. All right, so most of the talk today is going to focus on this path, liver to GI. But just so we go back and remember, what these parts of our diagram look like, this is just our general circulation of toxins. If things are going well, we can drain them out through liver and kidney. Now, when we block this path out of the liver, and we're going to focus in during the talk, we're going to go down and look at a hepatocyte level at what those transporters look like. But remember, when you block that path out of the liver into the GI tract through the bile tree, you dump stuff back into the blood. When that's blocked, it's called intrahepatic cholestasis, as opposed to extrahepatic. Extrahepatic would be blocks in your gallbladder. Intrahepatic is a failure of the transporters to move bile salts and toxins into the bile tree. Here we have enterohepatic circulation. Remember, we dump into the GI tract, but we reabsorb. This is one of the major reasons that we need toxin binders. And Then we have endotoxin. Endotoxin comes into the system. What's our biggest sort of nationwide, our biggest source of endotoxin as a dysfunction? It's leaky gut syndrome. So we have a lot of leaky gut. People talk about SIBO. Raise your hand if you think you have SIBO. Oh, yeah, right. Like half of you have been Googling SIBO for the last 2 years and thinking, oh, I got SIBO. All right. Why do you get SIBO? Because you don't have enough detergent in your upper GI tract. You are not flushing the upper GI tract with adequate bile. And so you have an overgrowth of bacteria, overgrowth of fungus. They are causing inflammation in the upper GI, causing a leaky GI, a leaky gut, and that's getting endotoxin into the system. Endotoxin is further aggravating all symptoms by increasing inflammation. And inflammation is a driver of toxic effect. It takes a toxin and makes it, multiple fold more toxic. Now, this idea of endotoxin and inflammation increasing toxicity is not really new. I've been talking about it for a while. Then I found this paper from 2001. And what they describe, if you just look at the title, you see what they're talking about. Concurrent inflammation as a determinant of susceptibility to toxicity from xenobiotic agents. Determinant of susceptibility. Inflammation is causing susceptibility. It is turning down the glutathione system, your chemo defense system, and then the toxin hits the targets much more acutely. And they describe pulses here. You see these spikes? These are pulses in endotoxin into circulation during different events. And when those pulses come in, there's an increased susceptibility to your toxin load. When those pulses aren't there, you might be fine with your toxin load. And then when the endotoxin comes in, suddenly that lowers your defenses, increases your susceptibility, and the toxins get you. And they have GI disturbance there. That's leaky gut. You get food poisoning, you induce leaky gut temporarily. Then they have alcohol. Alcohol induces leaky gut. Then they have an infection, altered diet, alcohol again, surgery. These are all pulses of susceptibility. So here they're talking about endotoxin. Now, let's go into experimental work where they used endotoxin to turn down the glutathione system throughout the liver and the GI tract. And in this study, it's called suppression of drug metabolizing enzymes. Read that as detoxification. Suppression of detoxification and efflux transporters, that's transport out of the system, in the intestines of endotoxin treated rats. And this is just demonstrating, this is intraperitoneal injection of endotoxin, creating IL-6 upregulation, which is inflammation. And that was across the whole GI tract. And then you won't be able to really see what's going on here, but these are all pathways in the liver. And your key pathway out of the liver, I already said, is MRP2. And MRP2 is the third one in there, and it goes down about 50-60%. MRP3 stays up. Think about it as staying open. Think about it as closing the gate out to the bile and leaving open the door back to the blood, the pressure relief valve. That's what we're saying here. You're blocked out of the liver, you go back into the blood. And just so you know how good endotoxin is, here they controlled it, they compared it to drugs made specifically to turn off those transporters. And endotoxin was every bit as good as any drug they could make to shut down efflux, toxin efflux. Now, it's not like this is all really new. This is from 1979. The guys who were studying cirrhosis and different liver diseases. Here we have contributions of gut-derived endotoxin to liver injury. And we have endotoxin going from the gut and nailing the liver, leading to impaired detoxification and changing in the immune system. And then extrahepatic effects, you have increased toxicity on the kidney and the heart, and they also have here the induction of fever because this is endotoxin inducing inflammation. All right. So now we're drilling down. We're in the hepatocyte. We're looking at the doors and the directionality of the hepatocyte. So on the left, you have the blood. On the right, you have the bile duct. That is the direction you want to go, from blood to bile and out. Now, what are the different doors? Over here on the left, you have OATP. This is bringing free toxins and conjugated toxins. Remember, in the cells in your body, you're conjugating mercury and other toxins to glutathione and shipping them out. And in the blood, you have unconjugated and conjugated toxins. They're both being pulled in by OATP. These are bile salt transporters that are taking enterohepatically circulated bile into the hepatocyte. And down here, you have the relief valves. That's MRP 3 and 4. 3 is for moving toxin conjugates out. 4 is for moving bile salts out. Bile is a detergent. It is an important detergent because it washes our upper small intestine. It washes out bacteria from the upper small intestine, and it's also, we know it as digesting or emulsifying our fats. But if it builds up in the hepatocyte, it emulsifies the hepatocyte. It kills the hepatocyte. Similarly, if the toxins build up, they can kill the hepatocyte. But here we're in a happy hepatocyte. So the toxins are coming in. Conjugated toxins go from the OATP right over there to MRP2 and get dumped into the bile stream. Unconjugated toxins go through phase 1, phase 2 conjugation, and then get dumped into the bile stream. And there we have BSEP, bile salt export pump, pumping bile salts into the canalicular membrane. And so all of that is going well, and this right directionality into the bile ducts. And there's one other transporter that's important to us, the MDR. The MDR is taking phosphatidylcholine. Same thing we supplement for liver and for cholesterol issues and for brain. It's what we use to make liposomes. It is being continuously harvested by the cell, from the cell membrane and put into the bile stream, where it joins with bile salts and makes mixed micelles to keep the detergent bile salts from digesting the lining of the bile tree and the gallbladder. In doing so, it is fluidizing the bile. One of the easiest ways to create cholestasis is not have enough phosphatidylcholine. Another way to create cholestasis is by blocking these transporters. So right directionality is movement into the bile. So now let's move into an endotoxin inflamed hepatocyte. In this is fascinating. For a guy like me, it's fascinating. Those transport proteins that were dumping everything into the bile actually get pulled out of the canalicular membrane and internalized in a little liposome-like structure. During a high endotoxin stress, you can destroy these proteins, and it takes a lot of energy to make them. So you move them internal and protect them from this stress. And look which two go together, the BSEP, the bile salt export pump, and MRP2. And MRP2 does not just move toxin conjugates, it also moves bile salts. So BSEP and MRP2 are together moving bile. MRP2 is also moving toxins, and they are together brought back into the hepatocyte when there is endotoxin-based damage. And what else happens then is the OATP, in the previous one, I had like 3 OATPs pulling stuff in. They get downregulated, and you make multiple copies of MRP3. And in extreme situations, MRP2 is taken and reinserted over here. The whole reason for all that is to turn up efflux back to the blood, because there is no path into the bile tract. And so you're forced to dump it all back into the blood, where at least it can dilute through the whole organism. In the context of detoxification, what are we going to see then? We're going to see skin expression, rashes. We're going to see lower back pain. As who tries to pick up the slack? The kidneys. We're going to see increased neuroinflammation, increased anxiety, increased brain fog, increased sympathetic autonomic tone, because we have failed to move in the proper direction. And just to show you that there is lots of data on this, this is endotoxin turning OATP way down. This is endotoxin turning MRP2 way down, and turning down, that's a bile salt transporter that brings bile in. So under endotoxin inflammatory stress, you stop bringing things from the blood into the hepatocyte and dumping them into the liver, and instead you dump the liver back into the blood. So this is a bummer. Then you end up at this situation. Your liver's screwed, your kidneys are impaired, and your brain is impaired. And just so you know that this all goes on in the kidneys too, this is a great paper, Augmentation of Mercury-Induced Nephrotoxicity by Endotoxin. So endotoxin taking one level of toxicity and raising it. And they have mice and they treat them with just mercury or just endotoxin. And mercury is a known nephrotoxin, so this is serum urea nitrogen. You see this little bump from mercury showing some kidney damage. Endotoxin alone, no kidney damage, that's control. Endotoxin plus mercury, add those together, you get synergistic damage. And so what happens to our excretion? There is urinary mercury in the mercury alone treatment, urinary mercury in the mercury plus endotoxin treatment. So we see a diminished release through the urine because we've damaged OATP and MRP2 in the renal proximal tubule. So we blocked kidney excretion. And where does that all go? This is whole body mercury, mercury alone, mercury plus endotoxin. So remember, it was only 1972 that Hal Huggins wrote about retention toxicity. He had a bunch of people in front of him with a lot of amalgams. All he could measure at the time was urinary levels of mercury. You got 5 guys with 20 amalgams. One's got really high mercury, one's got really low mercury in the urine. Who's suffering the most? The low. Retention toxicity. It was only once we were able to use mercury speciation to pull apart all these segments in all the compartments in the body that we could look at urine to blood ratios of inorganic mercury and show that. And here's a picture. Just remember, this is the unit of the nephron. There's glomerular filtration, and then there's the proximal tubules. All the transport's in the proximal tubules. Your GFR numbers may be fine, but your transport numbers for toxins can be significantly diminished. And everybody likes looking at SNPs, and we look at all these different SNPs. We have to get SNPs for the phase 3 transporters. And we have to be routinely running SNPs that deal with Nrf2 and deal with all the subsegments like glutathione synthesis, glutathionous transferase, and these transport proteins. Then we will be able to more accurately design our protocols for detoxification and more accurately know what to expect in each person. All right. So there's our messed up system. And what we're going to want to do here is clear out the blockage here, get everything flowing down and out, and trap it in there. So let's look at a proposed system for how we do this. Now, of course, there's got to be glutathione. You have to raise glutathione levels or support glutathione levels. There are nutritional ways to do that. There are also sort of shortcut supplementation routes like liposomal delivery. I'll show you just a little bit of data on what these deliveries can do when they're adequately done. Nrf2 upregulation. I'm most concerned right now, we're talking about taking an impaired person. The only thing I want to talk about Nrf2 upregulation is the use of diindolyl methane for removing blocks to Nrf2. And then most of our time is going to be spent on opening up bile flow in the hepatocyte and then catching all the toxins. So glutathione, we know we heard a lot about it. One of the things I like to shine a spotlight on, and Boyd went and did it anyways, right ahead of me, is glutathione's effect on the immune system. And so these are all just conditions that lower glutathione levels and increase infection rates. When glutathione goes low, your immune system shifts the Th1, Th2 balance. Th1, your innate immunity, your ability to kill viruses and bacteria goes way down, and Th2 goes up. That's your allergic responses to things. And in fact, glutathione is used inside the cell and converted to nitrosoglutathione, which is a very strong antimicrobial. So it's important from an oxidative stress perspective, from a detoxification perspective, and an immune perspective to keep glutathione up. Now, just to mention liposomes, because I'm a liposome obsessor, the idea of liposomes came around in the 60s. And it was like, wow, this is going to revolutionize all this stuff. We're going to take insulin orally through these liposomes. Never worked. In order for liposomes to work, they have to be very well designed. They have to be in a very small size range, and they have to have the right membrane constituents. And when you do them right, you can get them absorbing right through mucous membranes. This is a schematic of it going through the oral mucosa into the capillaries in there. And we call things liposomes, but really there's liposomes, there's nanoemulsions, there's hybrid lipid nanoparticles, but all of them can work when they're done right. And this is what's called a CACO2 study. CACO2 it's a cell layer on top of a filter, and you can put any compound that you're examining its permeability in across the intestine. You put a compound on top, and you look for it to appear below. So these cells that are grown are intestinal cells, and it's a model for intestinal uptake. And this was a B12 study, and we were looking at different sized liposomes and different compositions of membranes and how they get into the body. Now, to just direct your attention to the right, this was a study done in the literature looking at sizes of liposome and their uptake in a CACO2 model. And the real uptake only started, this is about 97 nanometers and 60 and 50. In these real small liposomes is where the uptake was really happening. And when we got a research group in Florida to do this for us, we examined size. So here's the control, and this is a rate of uptake. So the higher you are, the more uptake you have. And this is 130 nanometers, 80 nanometers, and 50 nanometers. So as you go into these small liposomes, you get very high transmissivity. And that was, essentially what they saw. There's their 50 nanometer, and that's about a 90, and that's 100. So they saw a similar decrease like that. As the size goes up, the permeation goes down. And in fact, at 138, it was no different than the control. And these were 3 different membrane compositions, all with a 50 nanometer liposome. And this is a surface modification that makes the membrane a little bit more water-like. And if we then take those same products and put them into somebody, you will see this is blood uptake of the B12. And what you see here is really rapid uptake. This is a 2-minute point, a 6-minute, a 10-minute, a 25-minute point, and a 50-minute point. So there's very rapid intraoral uptake. And this is the 50 nanometer particle with the surface hydration technology. And that in green is the 50 nanometer without that technology. And in orange is just B12 and water. So these things can work, and they can work for glutathione. This is the most exciting data I've seen in the last year. And this was from a group of Japanese doctors that work with us in Tokyo and the surrounding areas. A guy named Iso Kimura curates this group there. And they took 10 people, and they split them into 5 and 5. They did glutathione, blood glutathione by doctor's data before and 6 hours after either an IV of 600 milligrams glutathione or 600 milligrams glutathione in a liposome. Now, right after the IV cohort's going to be really high. But where does it go? How does it distribute through the body? How does it integrate into the biological system? Because glutathione is not meant to be in the blood. It's not a transport thing. And you have to take it apart and put it back together inside a cell. But some of the early data around liposomes showed that the ability for the cell to integrate glutathione was 100 times better in a liposome than it was in just IV. So at the end of 6 hours, what do you get? From IV, you get a 15% bump in blood glutathione, and from liposomes, a 30% bump. Anytime you get a 30% bump in glutathione, you should be jumping up and down. That's a pretty good thing to get in a single dosing. And so the liposome had a better interaction and a longer play through the system. So there is a way to get glutathione into the system now. With phase 2, the only thing I want to mention now, there's a lot of things to upregulate phase 2. Most of them are Nrf2 upregulators. Sulforaphane is the strongest. Very difficult to get sulforaphane active into the body. Diindolylmethane is one. I like that for a couple of reasons. And lipoic acid. I use a lot of lipoic acid. But when people are sick, they often have blockages to Nrf2. And I think that the next round of research that's going to be done, and it can be done on the skin 1 in the C. elegans model, is to see what things block Nrf2. Mold toxins have been shown to see it. We have a mold toxicity epidemic. We had a energy crisis in the 70s, and then we learned to insulate the heck out of all of our buildings so that nothing ever dries. And now we have a mold toxicity issue. And mold toxins can block Nrf2. And those people, you give them lipoic acid and they do bad with it. We had a lecture on hormesis yesterday. You think lipoic acid is an antioxidant? No. It's a little free radical generator in your cells, generating a mild free radical stress that is the Nrf2 upregulator. Polyphenols. Free radical generators. Sulforaphane. Why is it the strongest upregulator? Because it's the most cytotoxic. If it doesn't help you, it kills you. So if your Nrf2 is blocked, it's not going to help you. It's not going to make you feel good. So one of the interesting pieces of work that we were doing, I found this paper on DIM reversing Nrf2 epigenetic modification in prostate cancer cells. Everybody thinks about DIM for estrogen metabolism. But in the prostate, the cancer epigenetically blocked Nrf2, which means you can't detoxify, which explains all the old German research finding toxins in cancers. And they said that toxins created the cancers. Cancers block detoxification and accumulate toxins. So DIM was able to reverse this. So we used this on some of the mold patients, and on a good number of them, at least half, it was really miraculous. It really opened up a blocked liver. So I'm a big fan of DIM for that. DIM also is immunomodulating. It lowers Th2, Th17 hyperreactivity and makes the cells more T regulatory dominant. And so it's calming the immune hypersensitivities. All right, so now to the liver support. Let's go back in time to when the cure-all was bitters. Now, part of the reason that people love bitters is that it was high alcohol, and they would let you drink it as a medicine. And so there'd be bitter bars. Everybody would be jamming down bitters to get drunk. But why do bitters work for so many things? Because bitters turn up bile flow. Now, it was originally thought that it's working on the vagal nerve response as bitters touch the bitter receptors in the mouth, and that lets the gallbladder down. But it turns out there's molecular targets all over the place for bitter compounds, including upregulation of those bile salt transporters. And while we thought there was bitter receptors just in the mouth, then we found them in the stomach, then we found them in the pancreas. So the pancreas is tasting your food, and then we found them in the ovaries. Women who have trouble cycling, have difficult cycles, do extremely well on bitters. And, you know, some of the old naturopaths and Chinese medicine guys, these sort of crossover guys that taught me early on, said that gallbladder has, direct relation to hormone cycling, and women love being on bitters. That's Why? Because it's flowing the bile, and there is an interaction at the ovary level as well. So things for like gentian. Myrrh is an underutilized bitter. It's the major bitter detoxifier in Ayurveda. I've started using that in addition to the Western ones like gentian and dandelion. Oops. It is working on these transport proteins. In fact, in myrrh, there's glucosterones that are also present in the tree resin Google, not the web browser, and they upregulate bile salt export activity. So here we're tuning that to move out of the hepatocyte into the small intestine, and we're also, through the vagal response, affecting the gallbladder to release bile. So those are the main ones I like to use: myrrh, gentian, dandelion, solidago. There's many others that can be used as well. But that combo right there, if you just got tinctures and put them together, go a little low on the myrrh. It's really intense, and equal amounts of the others. The other thing is phosphatidylcholine. So there's a picture of phosphatidylcholine. The MDR is moving phosphatidylcholine to keep the bile fluidized. Without phosphatidylcholine, you immediately go cholestatic. And I always like to show membrane pictures because I'm a big membrane fan. And phosphatidylcholine is one of the dominant phospholipids in all of your membranes. And here are pictures of, there's the endoplasmic reticulum. There's another picture of it. There's the mitochondrial membrane. That was the same picture that Boyd was showing. And so that electron transport chain's all going through membranes. Most of the hormone transformations from cholesterol out to sex hormones to glucocorticosteroids, they're all in the membranes here of the endoplasmic reticulum or the membranes of the mitochondria. And it is the charge separation, the polarization of the membrane that drives these enzymatic reactions. And so when you're taking PC, you're feeding the system. But this is my favorite diagram of them all. Here you have the nucleus. Surrounded by the yellow is the endoplasmic reticulum. You've got mitochondria in green and the Golgi apparatus over here in brown, and they're all talking. The membrane-based organelles all communicating together about the potentiality in the cytoplasm, what can happen, what cannot happen, transmitting all that information to the nucleus to decide what genes we can express and which ones we can't. So we need a good intracellular and extracellular communication system for our body to work as a whole to handle all the stressors. All right, so I mentioned that bile is a detergent and cleans and clears the upper GI tract. And this is a very cool paper. I knew this must be true. I always talk about things before sometimes I do. I get all excited. I'm like, I know this is true. And this is a paper looking at small intestinal bacterial overgrowth, that's SIBO, in patients with progressive familial intrahepatic cholestasis. So they have a genetic predisposition to intrahepatic cholestasis. That's the one we're talking about, where you're not moving the bile into the bile tree. Those people are much more prone to SIBO. And when they treated them for SIBO, it came back. Why is that? And here is another from the web, this guy, Dr. Jockers. He lists the 4 major functions of bile: fatty acid metabolism, excretion of waste products, detoxification, kill off bad microbes, and blood sugar metabolism. So the detergent is cleaning. Once we break that through and get the bile in, it's cleaning out the upper GI. Bile flow is super, super important. All right, last thing to talk about is binding up. So we're getting the flow back out. We're getting PC and we're getting bitters. We're supporting glutathione, and we're getting the toxins out of the liver through the bile into the upper GI. So if we took this broken system, we open all that up. Boom, we open it up, we dump out of the liver, we go into the GI, and what happens next? We suck it right back up. Certain toxins are already prone to this, but we're usually working with a compromised system that's got a lot of leaky gut, so it'll have a higher proportion of things going in. And with that leaky gut, it's got a lot of endotoxins still coming in. So what do? We send our binders through, they come along, they march along, pick up our toxins, keep the toxins from going back in, keep the toxins from irritating the GI lining, and binding endotoxin and several dysbiotic toxins before they're ever even absorbed. And we'll look through a couple of slides with papers on the great ability of the simple charcoal to bind endotoxin. Remember, endotoxin is like gasoline on the fire. So, I like cocktails. All my friends know that? So my binders are no different. I want a cocktail of binders. Why do we want a cocktail of binders? Because we got a cocktail of toxins. Remember, it's not just the mercury world. It's not just mercury and fluoride. There's plasticizers, there's persistent organic pollutants, there's pesticides, there's herbicides, there's endotoxin, there's mold toxins, there's toxic algal bloom toxins. And every one of those has a little bit of a different character to it, its own chemical personality. And every one of the binders has its own chemical personality that's able to attract the toxins that are within its domain for binding. So we need a cocktail of different binders to get this cocktail of different toxins. So thiosilica, that's a silica particle saturated with sulfhydryl groups. There's so much that a gram of this thiosilica can hold 200 milligrams of mercury. So that's a great binder for mercury, arsenic, cadmium. Then we have charcoal. Gets a lot of different stuff, especially mold toxins and endotoxin. Clay kind of rounds that one out and gets a different subset. Chitosan is very interesting. It's a molecular mimic for Welchol, which is used in the Ritchie Shoemaker protocols for toxic algal bloom toxins and mold toxins. And then we use a lot of acacia gum and aloe. Acacia gum is a wonderful prebiotic for bifidobacteria, and it helps soothe and repair the lining of the GI tract, as does aloe. In the intestinal binders, let's skip those. That's more than we need to know. It's just, there's a lot of different toxins. Each one of the binders has its sweet spot, its mechanism. Put a bunch of them together, you're going to do way better than just one alone. And so of the different binders, there's the thiosilica. This is our original move. When I got into mercury toxicity, I dumped tons of DMSA into my body to try to get it out through my kidneys, and I kept measuring the urine, and there's no mercury in my urine, and I'm just spinning all this mercury around, jamming it into my inflamed kidneys, jamming it into my inflamed brain. And then I was watching Bob Roundtree give a talk on the GI tract and immunity, and I was sitting there in the depths of the darkness of my biochemical soul, the dark night of the biochemical soul. I was just ruined, and I said, oh, maybe I shouldn't go through the kidneys, maybe I should go through the GI tract. And I went back to my lab, and I cleaned up this stuff that I have that I make to pull mercury out of water, and then I started eating it, and it fixed me right up. And so that was the genesis of this whole trip here, is to get metals out through the GI tract and spare the kidneys. Just a little bit of the research on the other stuff. This is activated charcoal binding endotoxin. All right, endotoxin's one of the chief henchmen in this story, so let's bind that one up. But this is really interesting. Charcoal as a cytokine mop. And cytokines, pro-inflammatory compounds from your immune system, are causing a lot of this damage. Charcoal can actually mop them up, which means they must have some enterohepatic circulation. And this study, they gave cerebral malaria to mice, and this is the mice without charcoal and how many days they died in. From the time they were injected, they died in like 4 days. And if they gave them charcoal, they still died, but it took them 20 to 25 days because they damped down the inflammation. Oh, this is more just on clays and charcoals absorbing mold toxins. This is on clay absorbing mold toxins. This is similar, clay dampening down inflammatory reactions. We need to dampen that down while we're cleaning this system out. This is chitosan. It's derived from shellfish. It's deacetylated. The chitosan, it is non-allergenic. So it turns into this form. This form has these repeating amine groups that have a net positive charge, a partial positive charge. When it's slightly acidic, they have a fully positive charge. It's similar to Welchol. It absorbs anionic toxin metabolites that come out in the bile. All right, so we talked about toxin pathways. And remember, the liver, it's got to pull from the blood, dump. Pull, dump, pull, dump, pull, dump. When it gets too crazy, can't dump anymore, starts getting all frenzied, and goes right back into the blood. That's what's going on. We need to damp the endotoxin, damp the inflammation, stimulate bile flow, give phosphatidylcholine to get that open, and you will drain all these toxins, and then you got to catch them in the GI tract before they come back in. And if you have a really good delivery system that gets fast intraoral delivery, you can take all your Cholegogues and your glutathione system support, and 30 minutes later, you dump the binders in, clean up the whole mess. You just had a discrete cycle of detoxification. Dump, catch in 1 hour, and you're clean. You can do that once a day, twice a day, 3 times a day, as much as you want. But it's nice and discreet, and it's not leaving stuff back in circulation. It's not leaving stuff behind. So we looked at the pathways, we looked at the liver gallbladder, we looked at the binders, talk about glutathione supplementation, Nrf2 upregulation, a lot of ways to do it, but when you've got an impaired liver, think about DIM, diallyl methane, and then always Cholegogues and binders. All right, IAOMT, thanks so much. It was great to be back here again.